So hello everyone and welcome to our panel discussion, which will be looking at the translation of biomarkers and applications in the clinic. We're joined today by Richard, who will be moderating this session, and then our panelists are Abbas and Tanya. So over to you now, Richard.
Thank you, Esther. I think it's really an opportunity here to have this discussion regarding the, the biomarkers and the use also for, of course, in clinic, but also for the clinical trials that we have heard today, and has been the focus of this morning sessions, I would say. So I think it's great to have a representative from FDA, and actually specifically for the division that are looking into the qualification of the markers here. And also Tanya, as to my knowledge, you're also involved in the NIMBLE consortium, right? And I'm also very focused on the Litmus consortium that we are working on, trying to get some. So making Abbas time, using a lot of his time actually working with different kind of letter of intents, I would say. But I think, and I never had the chance to see the last slide of your presentation. So maybe you have to say that, as I said, on your presentation. But I think it's important to, when we're focusing very much on different kind of context of use. And right now, I think the field are really trying to have a surrogate marker to measure, because repeatedly measuring with biopsy is not really feasible, right? So we have that in our guidance, and we're really trying to find some good data to replace that. So even if most of our letter of intents are actually submitted on the diagnostic screening, we really want to go to the next step and try to understand what is the requirement for a surrogate marker approval. So if you can give little update on that, what kind of, can we have real world data, how many studies are needed, whatsoever, right?
Thank you so much, Richard. Of course, we get this question a lot. Everyone wants to go for that for the endpoint. And it's difficult. There is a guidance put out by the Division of Hepatology Nutrition on NASH endpoint biomarkers, and I think they've outlined what would consider would be considered as a start of for an endpoint biomarker, a reduction in the fibrosis score, and potentially the NAS score remaining the same or also decreasing. That would at least be the start of talking about an endpoint biomarker. To answer the other parts of your question, is real world data acceptable, and what kind of studies? Again, it's very difficult to answer the number of studies that's needed or the number of patients that are needed, but many times we are more than open to using real world data to support context of use and support the studies. Of course, when real world data is being used, something that becomes very difficult is, again, how consistent is the data, and how comparable is it to each other?
And I think you potentially using the liver histology data set as a reference, right? Mm-hmm. Because of course, if we never had the liver histology, we would actually go for hard clinical outcomes, and we might find another surrogate marker, right? So now we have the liver histology as a reference. And the question is, do we need to go through, do a new marker actually, obtain all the markers you have in the liver histology before they actually are approved as a surrogate marker? Or, how can you see that process? Because it's either, if we didn't have liver histology today, we wouldn't look in for NASH, we wouldn't look at that specific subgroup of endpoints to actually generate a new surrogate marker.
Right. Yes, you're correct. So of course, endpoints are usually associated with an outcome many times. So if you are going to, and I think there is a context of use that is more related to measuring outcomes, is a prognostic use for the litmus one, where they're trying to, the litmus LI, where they're relating it more towards outcomes. So yes, I do think outcomes is also something that, even though outcomes can be used, of course, that becomes difficult because it can take a long time to get to those outcomes or to show those outcomes, but outcomes is acceptable too.
Yeah, maybe what do you thought about that, Tanya?
No, I think it's an important point, right? And And we all, of course, want to look at actual outcomes in order for to qualify biomarkers, but that, of course, at the event rate that we typically see in these diseases, calls for a very, very long and drawn out study. So I guess another question, perhaps for you in the same space, is if there is longitudinally collected data that has some recorded outcomes, would that type of data set be acceptable for a prognostic COU, or do you think, and again, I know it's all devils in the details and the type of data and the quality of it, but do you think there is room to explore existing data sets, bank samples, which have some longitudinal outcomes and events associated with them as potentially a solution here, because otherwise we're looking at a very long drawn out study, right? So some thoughts or reflections on that would be helpful for us.
Yeah, you're right. I would say the devil, like you said, the detail. It's important to know the details. Again, we are, of course, open to looking at this information, but it's very hard. It's difficult to give a solid response without knowing those details. But that information can be useful in getting a better understanding, and maybe instead of having a larger number of samples later, like prospective samples, it may help with having a smaller number of samples as moving forward, or maybe, as you mentioned, not having to do samples at all if the data is very strong, the evidence is very strong, the sound and everything is very everything's very consistent, then yes. But more likely, I think it would be as helpful. It would be, I think, most likely, especially for endpoints, most of the times, it's important to have that high level of evidence for endpoints.
That's really helpful.
Are we using, when it comes to the data that you need to submit, it's one thing if you use a clinical trial data set, right? Then you can get a lot of details and you can play around with the data as you want in the authority, of course, when we share that data. But when it comes to published data, long-term published data, then do you expect that you can actually get the per patient level data, even if it's without actually personal information, or would you actually use the reference as a publication as your resource, right?
Yeah. This is again also a very good question. Publications are great. Again, it's really helpful to have that supportive information, but I would say most of the times we are interested in looking at that patient level data, to look at outliers, to look at the whole picture, which you really can't get in summary level data. So I do think it's very important to look at the patient level data for the final decision.
Yeah, that can be sometimes tricky depending on how that data are gathered, right? And also how the consents are actually done in that kind of study, in the registers and etc. Right? So that is something we need to look at. So that's good. But it gives us at least an idea. Then I guess. And I think that's actually what we both Nimble and Litmus have the opportunity, of course, to do this. We do lack, of course, long-term data, repeated biopsies. We do have something, but it's not, so that is, of course, what we're looking for, to try to gather and actually share that with you in the future. So I think that's good. But it's also, so what you say, it sounds like you're open for any kind of data set. It could be real-world data, it could be register data, it could be part of a clinical trial. So the totality amount of that would be, and also the analytical performance.
That's very key
done
too.
Within the study, right? Yeah.
Exactly. You're exactly right.
Richard, can I have actually question for Apas, which is a little bit of a clarification, and maybe others understood this, but you said that context of use can sort of evolve with the iterative conversations with the FDA a little bit, which I understand, and that's certainly been our experience as well. But of course, that the statistical plan in the QP stage, once it's locked, is locked, and that's sort of what has to be applied, right? Is that we just want to make sure that we are that I'm getting where is the flexibility and where is something that, as people are putting these things together, they have to be mindful of what they've committed to.
Yeah. So the QP is the plan.
Mhm.
So when you do send in your statistical analysis plan in the QP, ideally, it's not to say that it's locked in. There can be maybe some adjustments made. Like if you couldn't get the exact number that you needed, that'll be. I think that we could be flexible. That there's some flexibility in there, but essentially, you should be able to, even after the QP, show that your statistical analysis plan is going to support your context of use, support the patient population that you have. So what you plan for may not happen at the final FQP, and as long as it's explainable, it's still going to support overall the plan. That should be adequate. Of course, like things happen as you're going from the analysis, from planning to do analysis and doing the analysis, and that's understandable. Things can happen. That's understandable, but if you can't, I would say, like, you can't make a drastic change, and that would be where the plan's essentially now like out the window, or it's a completely different type of plan would be difficult
That's
to
right.
say like that. But yeah, so those kind of changes, some changes are acceptable because of course I think everyone's aware that things don't go as planned. So
Mhm.
No, it's good to hear that there is that level of flexibility and that openness to discuss as things evolve, and we learn new things as we are making our way through the biomarker qualification process. There's that openness to have that conversation and make sure that it's all in the service of the same end goal, and we stick to our plan. But there's some adjustments that are rational can be discussed. That's really helpful. Yeah.
So I have a follow-up question there. When it comes to qualification, of course, if we get something, a new surrogate marker for an approval for the FDA approvals, right? We still have this global complexity that we need an EMA approval and the Chinese CDE and the PMP, So are we, at least, do we have any collaboration or discussions over with EMA and FDA when it comes to qualifications part?
yeah, we do have meetings with EMA at certain occasions. We have them when there is something to discuss, especially when there are two projects that are the two identical projects are given at the same time. We will have discussions, and we want to be able to see what feedback they provided, see if it's if you want to provide similar feedback. Of course, support the use in of course America versus Europe and those kind of things. So we do have those discussions when needed, so that we can hopefully be consistent and have these biomarkers accepted more universally.
So you can call for a kind of a joint meeting.
Mm-hmm. Yeah.
Yeah.
Mm-hmm.
So good. And I think that's it's also, of course, the tricky, right? Because it's many stakeholders, and you also have the medical reviewers, that part of the discussion, right? And I think in the end, when it comes to qualification process for NASH, how much will the medical division compared to your division? How who's leading that process? Is it from CDER or is it from the the indication specific division?
No, when it comes to the biomarker qualification program, it is in the biomarker qualification program realm. It's not CDRH indication for use for like that regulatory process. So it's a biomarker qualification regulatory process, and the biomarker qualification program does work with with the CDRH reviewers and CDR reviewers, but the regulatory process is what's been laid out before the biomarker qualification program, the 21st Century Cures Act. So it's not a device. It's not going to be device specific. It's going to be more specific to the biomarker program.
Yeah, something I saw that from when I see from the letter of intent, the public information, right? That when, for example, artificial intelligence supportive readout of the histology, which will still need a histology, right? That seems to be moving from a surrogate marker context of use, question to a diagnostic screening. And I would say for a pharma industry, I would say we probably want to use the artificial intelligence more as a sensitivity analysis as a PD marker rather than actually a screening tool. So because then the screening tool we already there with the histology, right? So I think it's so the recommendation from the FDA there might not be totally in line with what we as the customer are having as an unmet need.
Right. So again, that's that was the initial feedback in the letter of intent, and of course, as as the submissions keep coming in and it goes through more review. As you mentioned, I think hopefully the context of use and the actual data will be will be able to be a supportive, more towards what's being used. And of course, exactly what you mentioned is there's no point in qualifying a biomarker for context of use that's not going to be used. We want to. This is very intensive on both parts. So we want to qualify a biomarker that's going to be usable and something that's going to be making a difference in clinical trials. That is, of course, considered.
Yeah, but that's why it's easier when you have this consortium, because then you have the pharma industry supporting any marker. We don't care. Anything that works, we are happy with, right? But as soon as you have one diagnostic company and an FDA discussion on the context of use, you lose out of one of the part that actually going to use it in the end. so I think that's also important that we get that kind of dialogue somewhere. So that's good. That's why we have these meetings. That's good.
I think I do agree. I think consortia-based approaches do tend to sort of, align across pharma, but also both Nimble and Litmus are working with so many diagnostic companies to make sure that we are qualifying biomarkers that can eventually go into how they are going to, apply for, sort of their device applications and things, which I think are completely separate, processes, but the same data set is usable and probably supports there. So hopefully we together can move through both the CDER and CDRH processes for qualification.
So is there anything from FDA that you want to recommend for us that we are sending in documents? Should we do that in a different way, or should we be more clear what we're trying to obtain?
No, of course, again, it's been phenomenal working with you and everyone who's in the space. We very much enjoy it. Enjoy seeing these biomarkers being developed and working on these biomarkers. I would say that there are, like I mentioned in my presentation, there are a lot of biomarkers that are being considered, and if the focus could be on to in some ways narrow down those biomarkers to biomarkers that actually that show the most promise, because again, it does take a lot of work. It is a lot of time and work on both sides to work on these biomarkers. So to focus on the biomarkers that show the most promise, whether it be individual or combination, would be probably the best. I think the best something to focus on too.
And then just, I think, to reiterate a point that was in discussion before, when you say biomarkers that show most promise, of course, analytically and clinically robust, etc., and usable, but also practicality of use. Is that a consideration? Are they actually deployable in a clinical setting? Yeah.
That's really important too. Again, we want to, when these biomarkers are qualified, we want them to be used. So if something, you're able to get that analytical data, clinical data for qualification, but then the biomarker is not used for the clinical trial, essentially doesn't serve a purpose. So yes, that's very important to know that it can be used and actually help get that patient population needed to improve NASH trials and get drugs approved.
Thank you. Any other guidance for us? I guess we have.
No, I think that's it. We're slowly working through guidance documents that'll hopefully be even more helpful. Will also be helpful. Hopefully these guidance documents will be completed next year sometime. But we are working on some guidance documents that will be out and will provide hopefully additional viewpoints to get that'll be helpful.
I think one of the confusions we have when we have this. De novo and the 510, etc., and we have the qualification process. I think the field, or when we can easily say that the ELF tests are FDA approved, and it's going to be in the drug development field, that then we can believe that we can use that as an enrichment tool. Of course we can do it because it's actually it's within the claim. But you still need a biopsy in the baseline and something to read out the effect of the drug. So, and that's not that clear. It's not that easy to understand that until. So that's why I think it's also the need of qualification is also something that we need to make sure that people understand. and make sure that we, as you say, we focus on the right one and we get something qualified as soon as possible.
Right. Very much so. Yeah, no, I totally agree. And again, 510K PMAs, that's very much for device regulations. The biomarker qualification program, biomarker qualification is under different regulations and has a different viewpoint. So there is overlap, but there's a lot of differences in what's being considered when they're going through those regulatory and both scientifically and regulatory going through those submissions.
Will the process go faster if you have something that already is a 510K approved, or having a class 2 something similar for ELF? Will the qualification process go faster, do you think?
Yeah, I would say most of the times, yes. This is just from my opinion, that when the device or the measurement method has CRH clearance, of course, it helps tremendously, because then it's already, a lot of the analytical data information has been provided, has been reviewed by CRH. So it just makes it more acceptable that since it's already gone through that CRH process and that regulatory process, at least speaking in that part, that there's some level of comfort already on that device or the measurement method.
Okay, thank you. So I think it's time for actually wrapping up that. Thank you for the discussion. This has been really, really interesting. And I hope for a continued dialogue and also more approvals and more reviews coming up very soon, hopefully. Okay.
Hopefully, yes. Thank you.
Thank you.